Hello everybody, this is Kavya and welcome to yet another interesting video in CSIR-NET series. So today we'll be talking about the P subunit, which is comprising of the composition, structure and also the function of carbs, lipids, proteins, nucleic acids and vitamins. So there are 5 in total and I'll be dealing with proteins first. So now let's talk about the biomolecules, which are the carbon-based organic compounds which are produced by a living organism, which are primarily made up of carbon, hydrogen, nitrogen, phosphorus and sulfur. So these compounds together make up the 99% of a living organism. So now we can see that there is a large molecule and also there is a small molecules. So large molecules are called as the macromolecules, which are greater than 1000 Daltons. So these are polysaccharides, proteins and nucleic acids. So polysaccharides are made up of sugars, proteins are made up of the amino acids and nucleic acids are made up of the DNA, also RNA and the nucleotide bases. And these are again linked to ribose sugars and phosphate moiety. So now the small molecules are the sugars, amino acids, DNA, RNA and also the fatty acids, which will make up the lipids. But the lipids do not come under the macromolecules because they weigh less than 1000 Daltons. So now first we'll concentrate on the protein section. So proteins are the polypeptide chains which are made up of the amino acids which are linked by an amide bond. And now these amino acids, when we take a closer look, these amino acids are made up of by codons. Single amino acid is made up of a codon and codon is constituting of the 3 nucleotide bases. So only when 3 nucleotide bases come together can they make up a single amino acid. So now when we look at this wheel, we see that 4 nucleotide bases A, U, G, C are present. And so let's look at this example, methionine. So if it is A, U and G, then it makes methionine, which is also the start codon. So in the similar manner, what each codon is coding for is given in this wheel. So that is how the amino acids are made up of, and by elimination of a water molecule an amide bond forms which will turn it into a polypeptide chain, and these polypeptide chain in turn folds to give rise to a three-dimensional protein. So we have 3 stop codons. So in total we have 64 codon, and out of which 3 of them are stopping. So if we eliminate the 3 stop codons, so in total we have the 61 functional codons. So the 3 codons are, see, if there is a in the center, you remember it as amber. And if it is double A, it's ochre, and the remaining is the opal, and this is how I remember too. So when we look at the structure of an amino acid in general, we are having a carboxylic group, and then we have a carbon which is attached to this carboxylic group. So this carbon is referred to as the alpha carbon. So this alpha carbon is then attached to an amino group, that is NH2, and apart from that it is also attached to a side group. So it is written as R because side group is variable. And as we know that the valency of carbon is 4, that means carbon attaches itself with 4 functional groups. So the fourth one, which is the remaining one, is the hydrogen. Now let's delve deeper into the amino acids. So this is referred to as the alpha carbon. That is because whatever carbon is next to carboxylic acid, that gets the number 1 position. So that is alpha carbon. And then as because amino group is attached to the alpha carbon, it is also referred to as the alpha amino group. So in nature, mostly alpha amino acids are existing. But we see that sometimes even beta amino acids are also more prevalent. So I'll give you an example. So this is H and H to balance the valency, and this is nothing but beta amino acid, because in the second case over here we see that this is alpha carbon and this is beta carbon, first carbon, second carbon, and the amino group is attached to the second carbon, hence it makes it a beta amino acid. An example for this is beta alanine. And so this is a beta-alanine structure. Beta-alanine is present in the pantothenic acid, which is a vitamin. And this pantothenic acid is a compound present in acetyl-CoA. It's one of the components which is present in the acetyl-CoA. So over here the logic is A is present in B, B is present in C. So obviously A is also present in C. So A is beta-alanine over here. So beta-alanine is also present in acetyl-CoA. So now we will discuss about the acidic and basic nature of the amino acid. So when amino acid is having both of this group, it makes it easier to dissociate in water and also associate back. So having both of this group makes it amphoteric in nature. Oh, I have already written it here. So this is amphoteric in nature. So now we have two possible acidic groups in the amino groups, and that is first one, the carboxylic group wherein the proton is present, and this is reversible. So when a proton is lost, it attains this form. So there is a net negative charge on it. And the second one is NH3+. So this is also a reversible reaction. When a proton is lost, it becomes NH2 and H+. So this is one condition, and the second condition is COO-. So this compound plus H+ give rise to COOH, and the next one is NH2, this one I am taking, plus H+ gives rise to NH3+. So these two are the acidic groups which are possible in an amino acid. That is because what is an acid? Acid is generally the one which is capable of losing an hydrogen atom. So because COOH is capable of losing a proton, that is H, hence it is turning into COO-, an ionizable group which is carrying a negative charge. And the same is seen with amino group, which is has accumulated an extra hydrogen, so it has given up that hydrogen. So because it is able to donate a proton, it is also an acidic group. Now when we look at the second condition, because the COO group is able to accept a proton, it wants to accept a proton, this is basic in nature. So these two are the basic groups which exist in an amino acid. So now over here we'll talk about the second concept, that is charge on the individual functional groups at three conditions. The first one is when there is low pH, and when there is high pH, and the third one is when it is at the physiological pH, that is 7. So a Zwitter ion is an ion which is having both positively and negatively charged groups. So now when we look at the first case, that is at low pH, the first case, we see that the carboxylic acid is COOH. That means because it is still able to give a proton, this is acidic group. And the NH3, the amino group is in the NH3+, which is plus charge. That means it is also able to give it. Give what? Give the proton. And in the second condition, that is the Zwitter ionic condition, at pH 7, we see that the carboxylic acid has lost the proton. So it is in COO-. So that means this has turned into a basic group which is ready to accept a proton, and the amino group is as it is. So that means it is again acidic group. In the third condition, we see that carboxylic acid is as it is because it has already lost its proton. So this is basic group anyways, Pg, and the amino group has also lost its proton. So it has turned into NH2. Now this also has turned into a basic group. So in the first one we see that it is acidic group, hence it's a cation. And in this second one functional group is basic and the other functional group is acidic, so which makes it having both plus and minus charge, hence it has also a dipole which are separated by two opposite charges, and hence is also called as a Zwitter ion. And the third group is both basic and hence it is basic, also called as the anion, which is represented by a minus sign. This is cation plus. So now we'll see the individual charges. Because this is NH3 plus, it has a positive charge over here, and COOH, it has neutral charge on this functional group. Over here it is having a positive charge because it has an additional proton, and here because it has lost that one proton which it has become -1. So this has neutral charge because NH2, that's it. So there's no charge over here, and this is having minus charge. So the accumulated charge over here is anion is having -1, this zwitterion +1 and -1 cancel up, so it is 0 charge, and cation is having +1. So this also gives us another insight into the amino acid, that is the cation, it is having +1 charge at low pH. At low pH an amino acid is carrying +1 charge. At zwitterionic form at the physiological pH at pH 7, so it can also be from 5.5, the range, it can be a range to 7 pH, it is carrying 0 charge, that is it is in the zwitterionic form because both positive and negative has cancelled out. So this is at the physiological pH. Then at the high pH it is having a charge of -1. So now we'll talk about another important topic, that is optical rotation. So we talk about the optical activity of the amino acid. So amino acid exists in two forms. One is the L isomer and the D isomer. So the L isomer is most prevalent in the nature and the D isomer is less prevalent in the nature. So whenever in a compound if D isomer is present, it cannot be easily cut, that is digested by the enzymes, because all our enzymes are made up of L form. Because this will be a D form, there is no not mostly a enzyme which can cut the D form. So now when we are talking about the L and the D, we have to see that L amino acid. So amino group is present towards the left side and D amino group, it is present towards the right side. So it depends upon the presence where the amino group is present with respect to the alpha carbon. That makes it either a L isomer or a D isomer. Now let's talk about the optical rotation. When we have a light source, the light source contains numerous planes of polarized light. But when we fix a polarizer, only a straight line, that is a straight light passes through it because only one plane of light is selected. When this one plane of light is selected and it is passed through a sample. So over here we'll have three conditions. First condition will have an L isomer, second will have a D isomer, third one will have an L plus D isomer. So in the first condition, when there is L isomer, that is amino group present towards the left side, we see that from the 0 degrees the polarizer moves towards the left side with an angle of -90. So why is it represented as -90? Because when a light is rotated towards the left side, it is rotated towards the left side because light is levorotatory, which is represented by a small l, also by a minus sign, that is rotating the light towards the left side. But in the second case, when we have the sample which is filled with the D isomer, that is amino group towards the right side, we see that the light is bending towards the right side with an angle of again 90 degrees only, but the sign is changing. And this is dextrorotatory, which is having a small d sign and also a plus sign. So over here, the angle of the rotation is similar, 90 degrees only, but only the sign is differing. The direction to which the light is bent is differing. Over here, the light is bent towards the left direction, and in the second case, it is bent towards the right direction. But when we consider the third direction, what do you think might happen when there is a combination of both L plus D isomers? Yes, you have guessed it right. There is a nullifying effect. Because this is rotating towards -90 and this is rotated towards +90, the angle cancels and there is a 0 degree rotation. It not necessarily means that the light has not rotated, it necessarily means that both of the isomers have cancelled each other and there is no rotation. So this is also called as the racemic mixture when there is an equal quantity of T plus L. And this is also called as a racemate, a mixture of equal quantities of two enantiomers. So what do you think an enantiomer is? These two are enantiomers. Suppose we separate them by a mirror. These two enantiomers, the isomers, are mirror images of each other, but these are non-superimposable. So if I just bring this and put it over here, the amino group comes over here, the CO groups come over here. So these are the mirror images but non-superimposable. The mirror images but non-superimposable are the enantiomers. So now let's learn about glycine. So what does it do with the glycine? So here we see that firstly we'll learn about the chirality. When a carbon compound is there, we know that to attain valency, it attaches itself, it shares its electrons with 4 different functional groups. If there are similar functional groups on all of the sides, then this is called as non-chiral carbon. In amino acids, glycine has a special property, that is because it is connected with 2 hydrogens. One is the usual hydrogen and the other one is the R group. Even the R group is hydrogen, and hence because these two functional groups are similar, this is optically inactive. So this cannot bend a light, it cannot rotate a light on either direction. This is unfit, that is, it's optically inactive. Only other amino acids except for glycine, if they're having a chiral carbon. What is the condition for being a chiral carbon? That is, carbon should be connected to 4 different functional groups. Then only it's called as a chiral carbon. So glycine has the most flexible side chain, that is because it is a small group and also a hydrogen group which is having partial positive charge, and it lacks beta carbon. So there is only a single carbon and no beta carbon. So amino acids having asymmetric alpha carbon exist in 2 enantiomeric form, L and D. So because this is symmetric, this does not exist in 2 forms, but the others except glycine, others exist in 2 forms, that is L and T form, as we have talked about here. And this R is a functional group which can be anything. If it is H over here, that it will become glycine, and because these two are similar, this also will not have any optical rotation or optical activity.